second, you will live.
You can come.
You're looking very nice.
Have a BGM Foundation. So we are very happy to welcome you. Whatever.
Three, two, one. So you are live now. You can start your session. Over to you, sir.
A very good evening to you all and welcome for today's webinar. Myself, Dr. Vamsi Murthy, and I'm part of the organizing committee for this GI webinar. On this interesting Sunday, we have esteemed national and international faculty eagerly waiting to enlighten all of us on microbiota in health and disease. So may I request Dr. V.G. Mohan Prasad, Chairman, VGM Hospital, to start this exciting session.
Hi friends, good evening to all of you and welcome to this a fantastic webinar today on gut microbiota in health and disease. So this webinar comes from the VGM Foundation and also along with Asian Institute of Gastroenterology. I'm just grateful to Padma Bhushan Dr. Nageshwar Reddy for joining hands with us. In organizing a series of 4 webinars. This is the last of the 4 webinars. So we're just going to have a fantastic feast today. We have a great personality to moderate this whole session. I have a great pleasure in introducing Professor Rupa Bane. She is MD, MRCP, DTM from Asian Institute of Gastroenterology at Hyderabad. She's a consultant gastroenterologist and also a great researcher. Her primary areas of interest are luminal gastroenterology, including IBD, the ulcerative colitis, Crohn's disease, microbiome, as well as the GERD. And she has a big database, more than 4,000 patient database, which she follows up. And she has multiple publications in national and international journals. And she has contributed to book chapters. She's also visiting faculty for many international workshops and is one of the most sought after speakers in national and international conferences and webinars. She has published a book on advanced GI imaging and also recently edited a book on IBD in clinical practice. And more than that, she has recently initiated the IBD-EE Emerging Economies Group with 16 countries of Southeast Asia joining hands together along with Middle East to promote collaborative work on IBD in this part of the world. She organizes AIG IBD Summit every year, which is very fondly looked for by all participants. With this, I have a great pleasure in welcoming Professor Dr. Rupa Bane from Asian Institute of Gastroenterology, Hyderabad. I will also go ahead and welcome Rupaji. So I have also a pleasure in introducing the other speakers, and then we move on to the lectures and followed by panel discussion. So I have a pleasure in introducing the international speaker, Professor Bernd Schnabl. Professor Bernd Schnabl is from the Department of Medicine, Division of Gastroenterology in University of California, San Diego, United States. He did his postdoctoral research fellowship from University of California, North Carolina, and Chapel Hill. He is also a GE fellow from Columbia University in New York, and he has done phenomenal work in intestinal microbiome and liver diseases, functional consequences of dysbiosis and chronic liver disease. And there is a laboratory in his name called Schnabl Lab, which looks at molecular mechanisms involved in the progression of chronic liver disease. He has more than 125 research papers in many international journals and also 4 book chapters and textbooks. He's a great researcher, and if you need to listen to know about microbiome, he is the person. The other eminent speaker today will be Professor Vineet Ahuja. He's a professor of gastroenterology at All India Institute of Medical Sciences, New Delhi. His areas of interest are luminal gastroenterology, including IBD, PUD, and also GERD. He's got a lot of research projects which are on in peptic ulcer disease and 6 of them in IBD. And he has many international and national journal publications. His present areas of interesting areas of research are microbiota in IBD and how curcumin modifies them, and what is the role of fecal calprotectin in assessing the inflammation in the gut in IBD. And also he is doing a research on GERD in Northeast frontier, rural versus urban. The panelist today is also a very eminent panelist, Dr. Cyriac Philip Abbey. So Philip got his DM from Hepatology and Liver Transplant Medicine from Institute of Liver and Biliary Sciences, when he got the gold medal from the President of India, the former President of India, for academic excellence in hepatology. His areas of interest are autoimmune hepatitis, microbiome, and drug-induced liver disease. He, became very popular because of one of the 154 publications was on fecal microbial transplant in severe alcoholic hepatitis, which made the whole country look at this FMT in severe AH. He has also co-authored a book entitled Diseases of Pancreas. So with this introduction, let me request Rupa Banerjee to give a few words of introduction. And then I have a few slides to share with you on microbiome. And then we will move on to Professor Vineet Ahuja and Schnabl. Over to Rupa Banerjee.
Good evening, everybody. Thank you, Dr. Vijay Mohan Prasad, for giving us the opportunity. I think this is a very interesting area that new developments are happening. And I think microbiome alterations are now being correlated or associated with a large number of gastroenterological diseases. GI malfunctions, dysbiosis is known to cause inflammatory bowel disease. And I think the future, what we are going towards, is alteration of the microbiome. Alteration meaning once the disease has happened, whether we can alter the microbiota to improve prognosis, and also maybe alter diet, alter the microbiome towards a healthy microbiome which can prevent disease. So this is a very interesting area, and I think we are really honored that we have the world's top leaders in this area, leaders from the country. I think Dr. Vineet, Dr. Phillips are doing a lot of work in the area of gut microbiome, and I'm sure this is going to be a very exciting session. Dr. Vijay Mohan Prasad himself is here. He will share with us a few slides on the microbiome basics in health and disease. Over to you, Dr. Mohan Prasad.
So I will start sharing my screen now. How do I get to my slides?
Now you can open your presentations.
Yeah, my present. Okay. Yeah. Yeah. Are you seeing my presentation now?
Yes, sir. It is visible.
It's visible. Okay. Great. Okay. Great. So I'll just give the initial intro on gut microbiome in health and disease. So, okay. So there are more microbes in the intestine than even human cells in the body. There are millions of microbes per square inch on the human body. So 100 trillion friends you did not know you had. And Hippocrates was very wise when he said all disease begins in the gut. So let us get into the journey of the composition of human microbiota. We are really more bug than man. The humans as microbiomes, we have 10 to 100 trillion microbes in our human intestine. We never realized we have so many microbiome in our intestine, 3 million genes. So there's 100 times more genomic material from the bacteria and the fungi and the viruses or bacteriophages in our gut when compared to even our own genes. So, and the whole weight of the microbiome, if you take, could be about 2 kilos. and about 300 to 1,000 species of bacteria are there. So this is really a baffling figure, and 100 trillion bacteria, and 95% of our microbiota are in the gut, and the genes, 150 times the genes of bacteria, and the is to 1 is the human genome, which means so much of genetic material is inside our gut, we don't even know about it. And they're medically accessible and manipulable, and the viruses is to bacteria is 5:1. So we don't even know many of the viruses in our gut. So what about the distribution of gut microbiota? In the stomach, because of the acidity, the microbiome is very less. Whereas as you move down, slowly it starts increasing. So the organisms that can lurk in the distal duodenum or jejunum could be enterococci and lactobacilli. Whereas if you go down to deep into the ileum and towards the large intestine only, you get all other bacteria. So you have almost the stomach is void of bacteria except for politicians like Helicobacter pylori, which know how to survive in an acid milieu by providing an alkaline milieu around them. But it doesn't happen with most of the bacteria, they get destroyed. But as you move down, the count starts increasing as the pH starts becoming less acidic and almost neutral or alkaline. So what are the major bacteria phyla and the genera predominating in the human gut microbiome? You see the firmicutes, or 60 to 80% are firmicutes, ruminococcus, Clostridium, Lactobacillus, and Enterococci are the major, firmicutes. So we all, know because we keep treating, our patients with Clostridium, Lactobacillus, and Enterococcus. Not much of ruminococcus is used in the probiotic that are marketed. You have Bacteroidetes species, 20 to 30%. So Bacteroides, and then Prevotella and Xylanibacter. You have Actinobacteria, less than 10%, which is one of the important bacteria which is used in all probiotics is Bifidobacterium, which is an Actinobacter. And then you have the Proteobacter, which is Escherichia E. coli, and then Enterobacteriaceae. All these are the proteobacteria. So there are good bacteria, there are bad bacteria. So commonly, some of the good bacteria, Bifidobacter, E. coli, some of the good types, good subspecies of E. coli, Lactobacillus. And then, of course, the bad ones are the Campylobacter, Enterococcus, and Clostridium difficile. These are some examples, but they're much more than that. So what are the factors which can affect the gut microbiome? So starting from the birth mode, is it a cesarean delivery or is it per via naturalis? 2, is the infant breastfed or not? And what about the diets? Is it basically a vegetarian diet or a non-vegetarian diet? And does the person do exercise or not? And is there a disease, any other diseases that affect the intestine? Like it could be a celiac disease or IBD or things like that. And how about is he from the toddler to old age? And then of course the drugs. And then of course the geography. The geography varies. Yeah, Professor Schnabl is here. So the geography varies. So from country to country, depending on the culture, the food habits and everything. So there is a window of opportunity for microbiota modulation, starting from the prenatal factors to neonatal factors, mode of delivery, gestational age, to postnatal factors. All these can modify the gut microbiome. So I will stop here, and then I will hand over to talk to Professor Schnabl, because he is the international celebrity on gut microbiota. So he is with us. So we will now move on to Professor Schnabl to deliver his talk. I'll stop sharing my slides. Professor Schnabl? He's not joined us yet. Okay, so I think there is some technical difficulty he's facing in joining. So I'll just continue speaking a little more on the microbiota by the time he just joins us. So I'll again share my screen. Can you see my slides now?
Yes, sir. We can see your.
You can see my slides?
Yes, sir. We can see your slide.
Can you see my slides?
Hello, sir. We can see your slide. You can continue.
Yeah, okay. I'll continue until Professor Schnabl is able to join us. Okay. So what about the role of microbiota in health? There is a symbiotic relationship. An interaction between two organisms living in close physical association is called symbiosis, typically to the advantage of both, which means the human being benefits from the bacteria in the gut, and also the bacteria also benefit from the food that the human being takes. So these are important in shaping and maintaining immunity. So both the innate immunity and adaptive immunity are all modified by the gut microbiome. So that is the most important thing. So the human being provides nutrients and shelter for the bacteria, because we are lodging a big group of different microbiome in our gut. And the gut microbiome increases the metabolic capacity of the host by aiding indigestion of plant carbohydrates, milk products, and also provides 10% of calories from the diet. Otherwise, the 10% would be passed off as such into the fecal residue. So they provide 10% of calories. They also provide vitamins like B12, B2, K, and folic acid, and then protect from the colonization with pathogenic bacteria. This is called colonization resistance. A good bacteria prevent the bad guys from getting attached. So this is the most important, slide, which is the gut mucosal barrier. So the intestinal barrier is important in the normal development of immune system. You see here, there is the intestinal lumen. This is intestinal lumen. You have a stirred mucus layer, and then you have an unstirred mucus layer. This unstirred mucus layer is the one that protects the intestinal epithelial cells. The microbiome lie here in the intestinal lumen, in the stirred layer and the unstirred layer, and they don't get attached to the microvilli unless otherwise this stirred layer goes away, the mucus layer goes away. One such example, if somebody takes mayonnaise sauce, the mayonnaise sauce is known to corrode the mucus layer. So the pathogenic bacteria can get access to reach the microvilli or the villi of the intestinal epithelial cells, thereby producing a damage. So this is tight junctions are seen here, so that none of these bacteria can ever cross unless otherwise they know some paracellular pathways. So to cross, not all bacteria can cross. Even the bacterial products, all of them cannot cross. So what are called the M cells are there, which take up these bacteria or their products and present it to the, this is the lymphoid follicle area where the M cells are there. So they present to the innate cells. These are called the B cells and the T cells. These are called the follicular B cells, follicular T cells, you have follicular dendritic cells, all these cells are there. So these bacteria and the products are presented to these cells. So these cells just taste it, discuss among themselves what should be the reaction. Is it an enemy? Should we mount an attack, or we can ignore? So there are other cells which are outside the lymphoid patches. So lymphoid follicles. These are also the B cells, T cells, dendritic cells, and the macrophages. So these also keep interacting with all the cytokines and immunomodulators which comes from the plasma cells. So all these, the microbiota and their, products which are tasted by the immune system, and appropriate action is taken. If it is a healthy gut with good bacteria, they don't react. Otherwise, pathogen, they all will mount an attack. So the bifidobacterium can produce a short chain fatty acid. The other commensal bacteria, they just send their flagellin, which is a portion of their surface, and then the lipopolysaccharides, sphingolipids. All these can interact with different types of receptors and can send different messages. So these are called the pathogen-associated molecular patterns, PAMs. And the PAMs are recognized by the pattern recognition receptor called PRR bearing cells of the innate immune system and many epithelial cells. So the PRRs, or the pattern recognition receptors, are found on our epithelial cells. And then the pathogen-associated molecular patterns, the PAMs. The PAMs are small, usually small carbohydrate sequences or carbohydrate like amino acid plus glucose sequence or things like that. They're small sequences which can be taken up and presented to the innate immune system. So the PAMs are produced by the bacteria, the microbiome. They are recognized by the PRRs on our epithelial cells. So this is what happens. The commensal bacteria, when they produce all this and send, these innate immune cells, even the T helper cells are called the TFH cells. They are follicular helper cells. So the follicular dendritic cells, they have TC cells. So these keep interacting, and if there is a antibody attack which is required, so immediately what will happen? The B cell, they form the memory clone. So immediately they expand to plasma cells, and the plasma cell starts producing bullets. These bullets are the immunoglobulin A. These IgA goes and attacks all these microbiome and their products. Again, all other commensal bacteria and Bacteroides fragilis, Clostridium, all these can interact also through the paracellular pathways, or they are taken up by the cells and passed on to the immune system. So the microbiota stimulation leads to B cell switch to IgA, or a regulatory T cell induction, the Treg cells. The Treg cells regulate unabated attack or replication of the helper T cells. So there is, or suppressor T cells either. So these are regulated by the T regulatory cells. So the T cells get differentiated to the different types of cells. If you take a naive CD4 positive T cell, then it can either get transformed to Th17 cell, or a Th1 cell, or a Th2 cell, or a Treg cell. The Treg cells produce the interleukin 10, which dampens all inflammatory attack, IL-35, TGF beta, which can regulate the immune response. The helper T cells are the ones which fight infection. So they can control infection. And the Th2 cells protect against parasites. And Th1 cells protect against intracellular microbes like viruses. So once differentiated, each lineage secretes a special type of cytokines and performs a specific function. So for a naive CD4 positive T cell to become converted to a Treg or a Th17 or Th2 or Th1 depends upon what organisms are there in the gut. Okay. So the protective function or the barrier effect of commensal bacteria is very important because they compete and adhere to the sites of attachment at the breast border of intestinal epithelial cells. They compete for available nutrients. They produce antimicrobial agents like bacteriocins. All this will prevent the attachment and subsequent entry of pathogenic bacteria into the epithelial cells. So I will stop here at this stage, and then I will leave it to our two experts to go ahead and speak. And I will right now I'll stop sharing my slide. Yeah, I think Professor Vineet Ahuja is already here. So I would invite Professor Vineet Ahuja to take over and give his talk now. Can you have this? Dr. Vineet?
I can hear you. I can hear Dr. Mohan Prasad. Sorry, I was in another webinar. I got stuck.
I know. So, Professor Vineet Ahuja will be speaking to us on microbiome, the Indian experience. This is of paramount importance to us. So, I think he started sharing his slides. So, I'll just mute myself. Thank you so much. I think you can put that height there in the last column, Vineet sir.
Okay. Okay.
Yeah, height. Yeah.
Okay. So thank you, Dr. Mohan Prasad, and thank you so much for inviting me for this webinar. Now, I basically thought that since we had a international authority was going to come and talk about the microbiome, let me play the devil's advocate and talk about the overhyped aspects. But in any case, it's not that I'm going to talk about the overhyped aspects only. What I would say is that I would give you an overview right now in terms of at present about the field of microbiome to some in a way linked to like what is happening and what new is happening out here. So this is a line from a book Lives of a Cell, and I think this is a line which actually makes you feel a little bit, I would say, sad, in the sense that when you read it and it says that we are not made up as we had always supposed, but we are shared, rented, and occupied. And this was not written for microbiome. It was written for cell many years back. And to think of it, that it is so much applicable today for us that we, a large part of us is because of the microbiome. That is what people say. In fact, it is said that 99% of the genome in human body is because of the microbiome, and 90% of the cells are because of the microbiome mass which is there. But is that true? And that is something which made human being called as a superorganism. It was thought that human being it's not consists of only what we thought of as a human being, but it was a reservoir. And that reservoir consisted of various kinds of microbiota. And when you're talking of figures like 99% genes in the human body are microbial and 90% cells in human body are microbial, then obviously it was a shock for everybody. But if you go a little bit and read between the lines, then you find that this figure was actually developed on the back of the envelope calculation many years ago. In fact, if you look at it properly, the true figure is much closer to 1:1, with the balance slightly in the favor of microbes. So that is the first myth buster. So 3 questions, and I would focus on them. What is new in them? Who and where are they? What are they doing? How can you harness them? When we talk about who and where are they, first thing we have to understand is that there is no standard microbiome ecology that all healthy people share. From 2010 to 2015, everybody talked about that what is the definition of a healthy microbiome. And the definition of a healthy microbiome, which people concurred was, okay, rich gene diversity, high resilience, high number of firmicutes, high number of bacteroids, low number of actinobacteria and all. And subsequently, when large data came up, for example, American Gut Project data, which looked at 9,300 individuals, these are all very healthy individuals. None of them has any disease. And what was seen out here is what are the kind of gut bacteria out there. It was seen that bacteroids were the maximum. However, bacteroids was not picked up in 1% of the individuals. 1% of the individuals did not have bacteroids at all. Even though we show in rest of the individuals that bacteroids are the highest. There is variation of some bacterial species, 5% of the total in one individual, and another individual only 0.01%. So clearly, extreme caution has to be taken in interpreting results from fewer than hundreds of people. Indian microbiome, we have plenty of studies from North India, from South India, and what we find is that the group from Pune, Dr. Yogesh Shauche's group, they feel that it is Privetola which is the maximum. Whereas the group is in Delhi, which feels that no, Firmicutes is the maximum. However, if you again look at it properly, you find that when it comes to phylum, they're both speaking the same things. When it comes to phyla, firmicutes is the largest proportion of gut microbiome in Indian population. And when we come to genus, then Privatola is the largest proportion in Indian population. And in fact, what has been seen that the gut microbiomes, they obviously differ amongst human population. So you're not going to extrapolate the microbiome which has been seen in a European population onto an Indian population. What is the clinical relevance for that? Clinical relevance for that is that if you're going to look at microbiome manipulation therapies, if you're going to look at, let us say, live biotherapeutic products which are going to come up with, which are based on microbial products, obviously they have to be different for each geographical location. In fact, like one of our papers, which was also quoted as a milestone in human microbiota research by Nature, it showed that even amongst the healthy population, this we're talking of healthy population, no disease, but people who are living in different habitats, people who are living in rural areas, people who are living in urban areas, or people who were living, we picked up people who are living in Ladakh. Why Ladakh? Because Ladakh, we had not seen too much of inflammatory bowel disease. So we wanted to see that what is the kind of microbiome out there. And these are all healthy people. So we expected their microbiome to be very much similar. They had no disease. However, it was seen that urban people, they had a worse microbiome as compared to rural people or people living in Ladakh. In fact, the rural people, they had amongst the best microbiomes. And these are people living in adjacent areas near Delhi, urban area, then a rural area. It is not that they were living in far off forests and all those things. So this suggests that all these things suggest that microbiome regularly show a large degree of interpersonal diversity, even in the absence of disease. Like the American Gut Project showed you that healthy people, and even amongst the healthy people, the microbiome bacteroids was the largest in many, but in 1% it was not present at all. So if you are going to characterize a healthy microbiome as an ideal set of specific microbiomes, then that's not a practical definition. What are the four major groups we talk about? We talk about firmicutes, bacteroids, actinobacteria, proteobacteria. We say that if firmicutes and bacteroids make up around 90-95%, that is a healthy microbiome. That may not necessarily be true for everybody. Second simple thing is it the cause or effect? And I'm taking inflammatory bowel disease as a disease example. And this is true actually for most diseases. Because if there's so much of talk is there about microbiome, so one has to be very sure that if microbiome is leading on to disease, is it because it is the cause of the disease, or it is the disease which has led on to change of microbiome? It is a chicken and the egg story, which came first. But yes, from therapeutic point of view, obviously it is very important. If you get a clear-cut signal that microbiome is the cause of disease, then obviously all your energy would be spent on changing the microbiome because you have found a cause. It is something like that. If you find that, okay, there's a monogenic disorder, if you find that there's an IL-10 deficiency in a person, in a young child, then you do not give that child too many drugs. You straight away go for bone marrow transplantation because you know that you have to correct that gene. That's all you have to do. So similarly out here, this question is extremely important. I'll give you the example of IBD, but I'll tell you this conclusion is true for all diseases. And the conclusion is that for any disease in human studies, till now, conclusively, it has not been shown that microbiome is the cause of the disease. It may have been shown in mice experiments. It may have been shown in transgenic mice experiments. It may have been shown in humanized mice experiments, but absolutely not in human longitudinal studies that microbiome is a cause of a disease. Again, let's take the example of IBD. For example, this is from 2005. We have taken patients of amoebic liver abscess, and there at that time we aspirated the liver abscess, and that is amoebic liver abscesses which are totally naive. They had not been treated earlier. No intervention had been done. And the classical teaching has been that a naive liver abscess is always sterile. And here, what was seen was that Peptostreptococci was seen around 70%. This was done by looking at the marker, the 16S RNA marker, which is a marker gene analysis or a marker gene which is present in all bacteria. So if you pick up this gene and sequence it, you're sure to take care of isolate all the bacteria. And subsequently, when you sequence them, you can also put them in phylum and genuses. So even though you're not cultivating any bacteria, you're able to identify bacteria. And as this example shows, that you could actually demonstrate bacteria anywhere in human body. And subsequently, in gastric tissue also, a lot of bacteria have been demonstrated, although earlier it was thought that only Helicobacter pylori is present out there. In patients of IBD, and when we compare it to control somewhere in 2010, it was seen that, okay, there are certain genera which are much more common as in patients with IBD as compared to controls. Fine. This again shows that, yes, it could be an effect of disease. There's an inflamed gut. When there's an inflamed gut, the habitat of the microbiome changes. So the habitat changes, so obviously the character of the microbiome changes. So that does not mean that they are causing the disease. We also picked up patients of amoebiasis, not the liver abscess, but patients who had colonic amoebiasis, and looked at their gut bacteria also, and compared them with the gut bacteria in IBD patients. Because here our hypothesis was that this is a case of chronic inflammation. Amoebiasis is a case of acute infection and acute inflammation. Are we going to find any difference in signature? We found that there was just a gut signature of inflammation. The signatures were roughly similar, suggesting that no, microbiome out here probably is not the cause, is not the cause, but probably the effect. And you may have an inflammatory signature mostly. There is maybe another signature which could be the cause. So we picked up cases of IBD which were mild, moderate, severe controls, and also looked at their metabolites. In terms of metabolites, we looked at whether what is happening to short chain fatty acids, because obviously data is already there that short chain fatty acids are the one which are deficient in inflammatory bowel disease. Short chain fatty acids are acetate, propionate, butyrate. So if they are decreased, IBD may occur, or IBD may get precipitated. So clearly there was a signature that patients who had severe disease had less number of short chain fatty acids, patients who had in remission had a higher number of short chain fatty acids. But again, does it suggest that short chain fatty acids are the cause or the effect? No, this could again be an effect of the disease. And so on and so forth. So if I collect all this information together, there were distinct microbiome signatures in patients who had acute severe ulcerative colitis, patients who had mild ulcerative colitis, healthy people living in urban areas, healthy people living in rural areas, and healthy people living in high altitude areas. But again, it does not tell you that it is the cause of the disease. All it tells you that, okay, in all these, depending upon the situation, the microbiome may change. So probably if you move the microbiome or transform the microbiome in a way that it changes, the signature changes from acute severe colitis kind of microbiome to a healthier microbiome, the disease may also settle down. And this is the basis on which the whole science of microbiome manipulation therapy is based. This is the basis on which the science of probiotics is based. This is the basis on which the science of fecal microbial transplantation is based. Apart from that, a definite cause-effect relationship has not been shown. Second point, when you're looking at microbiota, you can pick up microbiota from the fecal stream. You can pick up microbiota from the mucosal stream. You can pick up microbiota from the crypt specifics. Are there microbiota near the crypt? We do not know. We know that there are microbiota in the fecal stream, and that is the basis on which most of the studies have been done, because obviously it is very easy to collect the fecal samples. That way you're not manipulating the colon in any way, and you can go on and analyze the kind of bacteria which are present out there. For mucosa-associated bacteria, obviously you would have to take a colonic sample, or if you're taking if looking at upper GI microbiota, you'll have to take a duodenal sample. You need a biopsy. So is there any difference out there or not? And are there another set of bacteria present for which not much information is there, but probably they may be the master regulators? Because is it not possible that fecal microbiota are a set of bacteria which are just in transit? They may not have so much influence. It's like being how close are you to the center of power? If somebody is very close to the center of power, obviously they would have greater influence. Somebody is fast removed from the center of power, they would have less influence. So which is the center of power out here in the gut? The center of power out here in the gut is stem cell, which lies where? In the crypts. So any microbiota which would be closer to the crypts, whether it is mucosa or crypt, is going to probably have a greater influence. While if you look at studies, most of the studies have been based on fecal microbiota. So I'll just show you some recent data unpublished from our PhD students, Manaswani and Aditya, which shows transmission electron micrographs of mucosal bacteria from patients with IBD and healthy controls. And this is the villi. These are the mucosal microvilli, and these are the mucosal bacteria out there. And subsequently, these are the mucus. This is the mucus layer, and what you find is a number of bacteria out there also. Subsequently, when Watson's study was done, it was seen that not only there are bacteria in the mucosa, this is a bacteria which is lying adjacent to the mucosa, but there are also bacteria which are lying within the crypts, establishing that there is a crypt-associated bacteria. Prior to that, there is one French paper which has also looked into it and found some evidence of crypt-associated bacteria. Then we looked at whether there was any difference between the microbiome structure in patients who have in fecal microbiota versus mucosal microbiota, and it was seen that there is a difference in not only at the level of the phyla, there's also a difference at the level of genus also. In other words, that you will find some bacteria which are much more common in mucosal microbiota and some bacteria which are more common in the fecal microbiota, establishing that those are two parallel streams. It is not the same stream. They are two parallel streams, and they may be having a parallel function also. So clearly, composition has been an overhyped aspect, and we need to move away from that. Till now, we have been talking about, okay, do 16S RNA, that is a gene, marker gene for microbiome. That is a gene which is present in every bacteria. So if you pick up that bacteria, sequence that bacteria, then obviously you would be able to make up all the kind of bacteria which is present. But no, that is not important. Who they are is no longer so important. And where are they? Amongst where are they? Probably mucosal associated bacteria is more important. Comes the next question. What are they doing? Is this more important than who they are? Till now, we have been focusing on who they are. What are they doing? How do you decide what are they doing? How do the microbiota communicate with each other? The microbiota are going to communicate with each other with the help of metabolites they produce or the metabolites they may modify. So are metabolites more important than the microbiome? So this is a gastroenterology study which came up around 1 or 2 years back. This is a follow-up study of fecal microbial transplantation, high-intensity fecal microbial transplantation done in patients with IBD, showing conclusively better results with FMT. This is from the Michael Camp's group, and this clearly showed that patients who had achieved remission, they had a different kind of composition of microbiota, but more importantly, the metabolic pathways had also changed. They had abundant short-chain fatty acids. They had more of secondary bile acid synthesis. That was not so in patients who do not have any remission, and in remission, obviously, there was a decrease in hemobiosynthesis and all, suggesting that metabolites are different. But that was for overall group. So we wanted to see in a single patient, what happens in a single patient. In a single patient, metabolite change following FMT picked up patients of ulcerative colitis and also patients and also the healthy controls who otherwise who were undergoing sigmoidoscopy. But for patients with ulcerative colitis, we looked at their microbiome signatures as well as metabolite signatures prior to fecal microbial transplantation, and then also looked at their metabolite signatures following fecal microbial transplantation to see is there any difference or not. So what we find is these are the patients and this who underwent FMT, and these are the metabolomics of the fecal samples. So this is the control group. Control group is the healthy controls where we wanted to see what are the healthy metabolites. Now, these are the patients of ulcerative colitis who underwent fecal microbial transplantation. After fecal microbial transplantation, when 8 weeks later, we looked at their metabolites, we found that they had not only changed, they were moving much more towards the healthier controls, suggesting that patients who were benefiting, their metabolites were also changing. Clearly showing that metabolites actually are the doers. It's like the central dogma. Central dogma has been DNA gives rise to RNA, RNA gives rise to protein. It is actually the proteins which are doing the work. DNA is a driver. Similarly, out here, the central dogma would be, okay, the microbiome out there and subsequently the small metabolites and the small molecules. So it may be more important that what kind of metabolite is being produced, and that is what it shows. So the overhyped aspect of composition is no longer an important question. What is important is to look at the metabolite signatures. Now we come to how do you manipulate the gut, right? you have three choices. Although I would say if you really want to go way back in time, you would start with antibiotics, you would start with prebiotics, postbiotics, symbiotics. I'm not talking about them. I would like to talk about three agents, that is fecal microbial transplantation, diet, and live biotherapeutics, and I'm giving some snippets into them. Obviously, I'm not going to the whole science of any one of them. I'm just telling you that, okay, what is new which has occurred over the last 1 year. When we talk about fecal microbial transplantation, this is, I would say, this is one of the very interesting letters which came up in Journal of Hepatology. And I'm very happy to see that Dr. Cyriac Abby Phillips is also the panelist out here. And I'm sure that we would like to discuss this with him because it's a very interesting aspect he has raised. And the title of the letter is, as he has put it very aptly, he shows a flair for writing very well. And that is, you know my name, but not my story, deciding on an accurate nomenclature for FMT. In this letter, he argues, Dr. Phillips argues, right, that why not give it the name of intestinal microbiota reinstitution therapy? This is not transplantation. This is probably not restoration. This is just intestinal microbiota, and this is reinstitution. So this is an excellent piece of writing, I would say, an excellent thought process. Now, two sterling studies from Dr. Ajit Sood's group at DMC Ludhiana, where they looked at FMT, where they did randomized controlled trials of FMT and ulcerative colitis, and I've covered some very interesting data again. This is probably because till now, worldwide, we had 5 randomized controls of FMT in ulcerative colitis. But all these trials were for induction of remission in mild to moderate ulcerative colitis. So if you can induce for 8 weeks, that is it, full stop. You do not know what happens after that and how do you do that, whether you can maintain them with FMT or not. So this trial looked at maintenance of remission in patients with ulcerative colitis, a randomized control study where patients whom they had induced earlier with FMT, they randomized them into 2 groups, 31 patients, 30 patients, 61 patients total. One FMT plus the standard of therapy, other is placebo plus standard of therapy, and they gave them around 7 sessions of FMT in a year, followed them for a year, and although the primary endpoint of maintenance of clinical remission was not met fully, although the results in the FMT arm were much higher, the proportion achieving clinical remission was much higher, 87%, but these results were significant for endoscopic remission and histological remission. Very high proportion of them, 58% for achieving histological remission, 45% achieving endoscopic remission, 87% achieving clinical remission. And when it came to side effects, we had that scare of 2 deaths last year, which was highlighted by FDA about drug-resistant E. coli, which had been transmitted in patients who were undergoing FMT from recurrent Clostridium difficile infection. But this trial again, in a way, suggested that the side effects for FMT are not so serious, and if you monitor your donors very well, the side effects should be minimal. This group again showed that which are the best predictors of response to FMT, and they showed that age, disease extent, endoscopic Mayo score. These are the 3 factors. Younger patients, disease extent of E2, and Mayo score of 2 would predict a good response. And in fact, we recently completed a network meta-analysis of FMT versus biologics, which showed that for mild to moderate remission of ulcerative colitis, FMT seemed to be as good as infliximab or adalimumab. So that is again some very interesting piece of data. Regarding diet, I am putting up one study only because I think this study itself suffices the purpose in terms of if you want to get convinced that diet really helps. This study was done in pediatric IBD patients, a randomized controlled trial where they looked at Crohn's disease exclusion diet plus partial enteral nutrition, and they found that it induced sustained remission. In other words, they had 2 groups. In one group, pediatric IBD patients, in one group, they gave only exclusive enteral nutrition. In the other group, they gave Crohn's disease exclusion diet plus partial enteral nutrition. So this is the figure for this study. Left-hand side is their hypothesis. The hypothesis is Western diet or the habitual diet triggers Crohn's disease. Obviously, because it causes dysbiosis, increases intestinal permeability, leading to inflammation. So what if you do not change the microbiome, but you go a step higher? You go a step upstream. What changes the microbiome? What is the driver for microbiome? Obviously, one of the strong factors has to be diet. So why not change the diet? So they randomized the pediatric cases of Crohn's disease into two groups. One, they gave only exclusive enteral nutrition. Exclusive enteral nutrition would be like elemental or semi-elemental liquid formulation. It would not have any residue, and that would be exclusive. The patient would not be allowed to take any other food apart from that, but that would be complete in terms of calories, vitamins, minerals, and all. Other arm, they gave Crohn's disease exclusion diet. What is exclusion diet? Remove animal fat, remove wheat, remove dairy, red meat, emulsifiers, maltodextrins, carrageenan, and all. And add what? Fruits and vegetables. In other words, go back to the agrarian diet, the rural agrarian diet which India had, let's say, 50, 70 years back. Or if you go to remote rural areas, where people would be eating only fruits, vegetables, they may be having dairy and wheat, but definitely less of red meat, emulsifiers, maltodextrins, and all those things. And what it showed was that even though when patients were put on EEN, which is also very less immunogenic, despite that, not only CDED, that is exclusion diet, plus partial enteral nutrition was better tolerated than exclusive enteral nutrition. Both were effective in achieving remission at week 6. And in fact, the exclusion diet was superior and sustained remission and reduced inflammation at week 12, suggesting that diet is a very, very strong player. And not only is it a strong player, probably it would have the least side effects, and probably it's a physiological way of treating IBD. So with diet, whether you can treat severe forms of IBD. This month, we put in some data which showed that when it compared patients of ulcerative colitis with, again, very severe colitis who are hospitalized and healthy controls, you find that patients who have acute severe ulcerative colitis, the microbial diversity is the lowest. They're the least amount of alpha diversity, the Shannon indexes were low, and if you looked at individual firmicutes and bacteroids, they were firmicutes, Clostridium were low, and Bacteroidea were also low, suggesting that they have an inflammatory kind of microbiome which is out there. So now in patients of acute severe ulcerative colitis, doing fecal microbial transplantation obviously requires, it's a brave effort. You need a lot of courage, I would say, to do that. And obviously there is no worldwide data also on it. And at the same time, if you want to start a randomized control trial, you do need a lot of courage to do that. You're not sure whether it will help the patient or not. So we thought that if you want to restore the microbiota, and at that time, when the patient has acute severe ulcerative colitis, obviously there are limited dietary strategies you can try. Why not do a randomized control trial whereby you give them exclusive enteral nutrition? Exclusive enteral nutrition, as I've already told you, is maybe an elemental or a semi-elemental formulation. So we did a randomized control trial where 62 patients of acute severe ulcerative colitis were randomized into one arm, only exclusive enteral nutrition, plus the standard of care for acute severe ulcerative colitis. They were given steroids, if they had to be given antibiotics, as antibiotics, and enemas and all. And the other arm, the standard of care arm, but no EE. The patients received exclusive enteral nutrition, which was semi-elemental, for 7 days. And in addition, we also did day 1 and day 7 fecal microbiome analysis, because we were very sure that with exclusive enteral nutrition, whether we achieve any clinical effect or not, we will definitely be able to change the microbiome. So let us look at the results, what happened. So these are the results of biochemical and inflammatory markers. And what it shows is that proteins improved in exclusive enteral nutrition group. Obviously, they were given good diet. Albumin improved. CRP levels went down. Fecal calprotectin levels went down. And they all improved out here, and they went down out here significantly, suggesting that biochemically and inflammatory, there was definitely a very, very good response. What about clinical endpoints? Here I'm showing you per protocol analysis, because in intention to treat analysis, we just reached significant values. So it was seen that steroid failure was much less in patients with exclusive enteral nutrition group, only 19%. There was a delta of 24% between both the arms, and that is a big gap for patients with acute severe ulcerative colitis. 24% of patients, just with one intervention, they are avoiding steroid failure. In terms of duration of hospitalization also, it was less, although colectomy rates were similar, suggesting, I'm not saying that this is a definite strategy, it is just one trial, but suggests, and this is a per protocol analysis, that concomitant and exclusive enteral nutrition may augment steroid responsiveness in patients with acute severe ulcerative colitis. In other words, if you change the diet, it helps. As I told you, our a priori hypothesis, the second a priori hypothesis we had was we were very sure microbiome would surely change. When we looked at the microbiome, we were surprised. There was not much change in the microbiome. These are patients who were not given EN, these are patients who were given EN. Some change in species, but not much. This is not the kind of effect which we were anticipating, although we got a clinical effect for sure. We got a biochemical effect for sure. We got a decrease in inflammatory markers for sure. This shows that patients of acute severe ulcerative colitis, when they have a bad microbiome, it takes some time to recover. That microbiome is in a stage of exhaustion. It's a stunned microbiome, and it takes some time for recovery to take place. Finally, I feel that the field of FMT is soon going to give way to what are known as next generation probiotics or the live biotherapeutics. At the background is a table which shows around 10 trials which are already taking place, phase 1, phase 2, preclinical trials, whereby these are trials for live biotherapeutics or metabolite-based capsules. So what are live biotherapeutics? Live biotherapeutics is that you pick up the bacteria which you feel are going to help. And once you pick up those bacteria, culture those bacteria, make capsules of them, and then give it to the patients. But what you do is that you pick up the metabolites, make capsules of those metabolites and give it to the patients. Obviously, these are not as simple as doing FMT because FMT never went through the regular process of regulatory things and all this. Whereas here you are culturing organisms. If you're going to culture the organisms, it has to go through a proper regulatory process. That is a challenge and they may be expensive, but this is the way things may happen. And with COVID times, the way it has become a challenge at every place to do FMT because of COVID. So obviously, the future is going to belong to live biotherapeutics. So the overall aspects and overall, what I would say is that we have been talking about composition. When it comes to microbiome, composition may be an important question, but may be more of an ancillary issue and not the most important question. So studies which are targeting 50, 100 patients, looking at 16 sRNAs and providing the composition are good for publication, but they are not providing definite results in terms of translational value. Mucosal associated bacteria may be more important than fecal microbiota. And what is going to actually provide results for translational value is what those bacteria which change, what kind of metabolites change. The metabolite signatures are going to be more important. FMT obviously is having its, I would say, place in the sun in terms of that it is trials have started for graft versus host disease, trials have started prior to chemotherapy regimes, trials have started for various kinds of mucositis, Alzheimer's disease, multiple sclerosis, autism, leave aside gastroenterology and hepatology fields. But yet at the same time, there's a possibility that FMT is going to be superseded either by dietary therapies, as I showed you, exclusive enteral nutrition in acute severe ulcerative colitis, or the Crohn's disease exclusion diet in patients with Crohn's disease, or by the next generation probiotics, also known as live biotherapeutics. Thank you very much for your attention, please.
I think covering not only the overhyped parts, I think covering the facts as well. And I think this is the very exciting area that we are seeing in gastroenterology. So we will have a question answer session at the end of the talks. I think Professor Bernd, has he joined? Professor Bernd Schnabl? Yeah.
Yes, I'm here.
Yes, so it's indeed an honor to have you with us, and we're looking forward to hear your talk. Dr. Vijay has already introduced Professor Schnabl. I think in India also a lot of work is being done, as Dr. Vineet has just said, and we're looking forward to hear from you on microbiota in health and disease. A small talk on the side. I would request all audience, if you have questions, to please post it right away so that we can have an interesting panel discussion, and the lead speakers will be available to answer all your queries. Over to you, Professor Schnabl.
Yes. Can you see my slide?
Yes, we can see your slide.
We can see, yes.
Perfect. So thank you so much, first of all, for inviting me to talk about microbiome and health. I will, in fact, focus on predominantly liver disease. And in fact, I will also focus within the liver disease field, focus very much on alcohol-related liver disease and how can we translate this microbiome research into therapies for liver disease. These are my disclosures. This is also my overview. I want to initially introduce you, and I'm sure many of you know about alcoholic liver disease. I just want to introduce it a little bit, and then I want to specifically focus on the viral.
Hello, sir. Sorry to interrupt you, but your voice is breaking, sir. Can you remove earphone?
I'm not.
Sir, you are not audible without earphone. Sir, you are muted. Sir, you are muted. Can you unmute yourself? Sir, just open the site and unmute yourself because as of now you are muted. Yeah, now unmute yourself. Yeah. Sir, it's still breaking. Can you remove the earphone now? Sir, now you are on mute. Yeah. Hello.
Now?
Yes. Now clear, sir. We can
Yes,
hear
can
you now.
you?
Yes, sir. We can hear you now, sir. You can
Okay.
now on your presentation. Yeah.
Okay. So.
Thank you.
Okay. Is it better now?
Yes, sir.
Okay.
You are audible.
So okay. So again, these are my disclosures, and I will talk about the alcohol-associated liver disease and focus on the virome, the fungal microbiota, and the bacterial microbiota. As you know, this is the natural history of alcohol-related liver disease, the disease that I predominantly will focus on today. As you know, that if you have chronic alcohol abuse, eventually everybody will end up with steatosis, but the minority eventually will move on to develop more advanced liver disease like steatohepatitis, fibrosis, cirrhosis, and hepatocellular carcinoma. We have one special entity, which is alcoholic hepatitis. It can essentially arise in patients with any stage of liver disease. However, predominantly patients with alcoholic hepatitis have an underlying liver cirrhosis. The 90-day mortality is between 20 to 50%, and there is currently no effective therapy. At least in the US, we have alcohol-related liver disease is the leading cause for liver transplant currently, and it's also rising. And we heard already about the gut microbiome. I just want to remind you that the gut microbiome not only consists out of bacteria, but we also have fungi present. And in this case, in the intestine, we have about 1/4 are Candida, and then we have viruses as well. And from these viruses, we have predominantly bacteriophages. We have heard that we have about the same number of human cells as we have microbial cells, but the microbial genome outnumbers the genes that we have in our human genome. So does the gut microbiota contribute to alcoholic hepatitis? And I just want to show you one preclinical and one clinical study. The preclinical study was published several years ago, and Gabriel Perlmutter from Paris, he used germ-free mice. He colonized them with stool from alcoholic hepatitis patients and then fed these mice chronically ethanol. And as you can see here, after ethanol feeding, these mice colonized with the stool from the alcoholic hepatitis patients had more liver injury, more liver steatosis as compared to the conventional mice. So suggesting that, in fact, alcohol-related liver disease, at least in this preclinical model, is transmissible. We have Dr. Phillips joining us today. He published this very nice pilot study using fecal microbiota transplantation in steroid-ineligible patients with alcoholic hepatitis. This was done over 8 days via nasojejunal tube. And you can see here that the FMT group had a very good survival of 88%, versus historical controls had a survival of 33%. So suggesting that there is at least something also in the in patients, that this improves with exchanging the gut microbiota. So I want to show you briefly how the gut virome changes in patients with alcohol-related liver disease. This was just recently published by us. And what we did is we extracted viral particles from the feces from patients with alcoholic hepatitis, alcohol use disorder, and controls. And by this heat map, you can very nicely appreciate that the virome in alcoholic hepatitis patients is very much different from the alcohol use disorder and control patients. When we looked at the diversity, as opposed to what you will see later on with the fungal diversity and bacterial diversity, the viral diversity, in fact, decreases in the controls. So in other words, in alcoholic hepatitis patients, we have the highest viral diversity of all of these three groups. And as you can also appreciate already by these pie charts here, one of the significant changes that we found is that the mammalian viruses shown here in yellow are actually increased in alcoholic hepatitis patients. And what we found is that, for example, the two most significant changes were the parvoviridae. And again, this is in feces, this is not in serum. We had about 50% of the patients with alcoholic hepatitis and alcohol use disorder patients had parvoviridae present as compared to the control patients. And in particular, the herpes viridae, and this is particularly presented by EBV, so Epstein-Barr virus. We had about 15% only in the alcoholic hepatitis patients. We could not detect this in the alcohol use disorder patients and the control patients. And when we looked at the alcoholic hepatitis patients where we detect this herpes viridae, they had a significantly higher MELD score as the patients where we did not detect herpes viridae. And this is in particular interesting because this discriminates, and I'm not showing the data here, this discriminates the virome from patients with NASH and NAFLD. In NAFLD and NASH patients, we could not detect any expansion of the mammalian viruses. So this is kind of interesting and obviously requires some more follow-up studies. I want to switch over to the fungal microbiome. How does this change in alcohol-related liver disease? And when we initially did this, and this is one slide showing a mouse study, and we had this, we just recently published this, but we had this result present for many years now in my laboratory. You can see that this is almost a black and white image. So you have in the control mice, if you do just regular YPT plates to culture fungi, you almost have, most of the mice don't show any colonies, and then you have some mice that show very few colonies. And then if you feed them alcohol, it's all of a sudden you get this overgrowth of fungi in the intestine, in the feces from these mice. And we can also quantitate this by culture independent techniques, by qPCR. You can see that we get this fungal overgrowth in the mice. And then we also have this beta-glucan, which is part of the cell membrane from many fungi. you can see that we have also beta-glucan is present in the blood from these mice. And also, if we treat these mice with amphotericin B, so in this case, we use a non-absorbable amphotericin B, so we just gave it by gavage. Or in this case, actually, we mixed it into the diet. You can see that we can decrease the total fungal load, and also this beta-glucan we can significantly reduce. So how does this look like in patients? Very similar. So here we have the control patients, the alcohol use disorder patients, and the alcoholic hepatitis patients. Again, in the non-alcoholic controls, we almost cannot see any culturable fungi. And again, this is a very simple technique with the YPT agar. This is not like a very sophisticated technique to look for different species, but you can very nicely see that in alcohol use disorder, we get an increase in the fungal load in the feces or in the stool, and even more in the alcoholic hepatitis patients. And when we do a colony PCR, this is in the alcoholic hepatitis patients, it's predominantly actually C. albicans. When we now do ITS sequencing, so this is a specialized sequencing for the fungi, and in this case, we are using a conserved region in the fungi, which is comparable to the 16S region that we use for the 16S sequencing for bacteria. You can also appreciate that in the control patients, we have a very nice diversity of different fungi present. Consistent with published studies, we have about 1/4 is Canada. Excuse me. And then we have Canada essentially takes over in the alcohol use disorder patient and the alcoholic hepatitis patients, which is also quite striking. When we look at the PCA analysis, so how do the microbiota compare between these three groups? We cannot distinguish just by this PCA analysis the microbiome between alcoholic hepatitis patients shown in red and the alcohol use disorder shown in blue. But we can significantly discriminate the fungal microbiota from patients with non-alcoholic, from non-alcoholic controls. And as opposed as what we have shown you before with the viral diversity, in this case, the viral diversity decreases in the diseased patients with alcohol use disorder and with alcoholic hepatitis. So suggesting that this is the opposite what we saw with the viral diversity. However, we can also not see a significant difference between the alcohol use disorder and the alcoholic hepatitis patients. So suggesting that in fact this is due probably to alcohol use rather than to the liver disease stage. Finally, moving now over to the bacterial microbiota and showing you some of our more recent data that we have also published by now. How does the bacterial microbiota contribute to alcohol-related liver disease? We know from very old studies, this was published in 1984, when Bodhi actually took aspirates during an upper endoscopy from the duodenum and just cultured them. We knew that the anaerobic and the aerobic bacteria are increased in patients with alcohol use disorder versus the controls. And we have also confirmed that this is not only true in the lumen of the duodenum, but also the mucosa-associated microbiome is where bacteria are increased. So in this case, we have a 5-fold increase in the mucosa-associated bacteria in patients with alcohol use disorder as compared to the controls. We obviously wanted to know how does this change by using 16S rRNA sequencing, and we used a control group, again, alcohol use disorder patients and alcoholic hepatitis patients, and just by the PCA analysis, you can see that the microbiota between these three groups is significantly different from each other. Now, when we look now at the bacterial diversity, this is also very similar to the fungal diversity, that in disease, the bacterial diversity decreases. However, we have a gradual decrease now. So we have a decrease of bacterial diversity from the controls to the alcohol use disorder patients with probably more mild liver disease. And then we have a further decrease in bacterial diversity from the alcohol use disorder to the alcoholic hepatitis patients. And one of the most significant changes that we found by 16S sequencing was this large increase in Enterococcus faecalis. We found about a 3,000-fold increase in alcoholic hepatitis patients as compared to the controls. Now, when we did a correlation analysis between either the absolute amount or the relative abundance of Enterococcus faecalis to the clinical outcome, to the disease severity or the mortality from these alcoholic hepatitis patients, we could not find any significant correlation. However, this was still interesting, this finding for us. We have published a couple of years ago that if you colonize mice with Enterococcus faecalis, very simply, you can in fact induce mild liver steatosis in these mice. You can also increase liver injury by ALT levels measured here. You can get more liver inflammation. And then if you move on and feed these mice ethanol, you can get more ethanol-induced liver disease, shown here by increased steatosis, more liver injury, more liver inflammation, and you can induce some degree of liver fibrosis. So the question was, if it's not the abundance of Enterococcus faecalis, what is the cause? Why is this inducing mild liver disease and exacerbating alcohol-induced liver disease? And is there anything also going on in our patients? So we scratched our head and, we said, what could it be? And we, it took us a while to get around this and to find what are these bacteria in fact doing. And what we discovered was a toxin which is called cytolysine. And when we measured cytolysine by qPCR in the feces from our patient and control cohorts, you can very nicely appreciate that cytolysine was not present in the control patients. We had one patient in the alcohol use disorder group that had that was positive for cytolysine, but after 2 weeks of abstinence, this patient also turned cytolysine negative. But then we found this, like 30% of the alcoholic hepatitis patients were cytolysine positive. And this was kind of interesting, and even more interesting when we looked at the disease outcome. Speaking about the mortality, you can appreciate that cytolysine negative patients survived these 180 days, while most of the cytolysine positive patients died within 180 days. And using a multivariate Cox regression analysis and adjusting for multiple parameters, including the MELD, but antibiotics use and steroid use, you can see that we have a hazard ratio of about 14. So this patient has a 14-fold higher chance of dying within 90 days if he or she is cytolysine positive. So the question is, is this only a good biomarker for disease, or is there anything else behind this? And to answer these questions, we obviously have to move towards preclinical mouse models. And before I show you the preclinical mouse models, I would just want to also emphasize that the cytolysine positive Enterococcus faecalis is in fact phylogenetically diverse, so it does not seem to be a nosocomial infection. This was a multi-center study from Europe and from the US and Mexico. And you can see that the percentage of the subjects that were positive for cytolysine was in fact comparable within all of our groups, between 20 to 50%. And again, this has nothing to do with any of the liver diseases, at least as far as we know by now. We have also published a study recently showing that in fact NAFLD patients with NASH or cirrhosis, they are also not more prone to be cytolysine positive. And there's also no correlation between cytolysine positivity and NAFLD disease or disease outcome. So turning now to what I've earlier mentioned, to preclinical models, we have to show, is cytolysine now exacerbating liver disease? And what we did very simply, we used wild-type mice, C57 black 6 mice. We colonized them either with a cytolysine positive Enterococcus faecalis strain. We used a isogenic strain that is now mutant in the cytolysine. And as a third group, we had the PBS gavage. And when we move on and subject these mice to the ethanol-induced liver disease model, you can very nicely appreciate that the mice that were colonized with Enterococcus faecalis, they had the highest degree of liver disease and liver steatosis as compared to the mice that were colonized with Enterococcus faecalis with the mutant cytolysine. And also as a control group, we have a PBS gavage group. So suggesting, in fact, that at least in this preclinical model, Enterococcus faecalis cytolysine increases ethanol-induced liver disease in vivo. So what is cytolysine? Cytolysine is a very small toxin, about 5 kilodalton. It has a small and a large subunit, and it is secreted by Enterococcus faecalis. Specifically, it's encoded by 2 different genes. If there is no target cell around, these subunits form multimers and they are getting inactivated. However, if you have a target cell around, these subunits can form what we currently believe is pores in the bacteria, but also in mammalian cells. So is this also true for the mouse liver cells, specifically for mouse hepatocytes? So we isolated mouse hepatocytes. We incubated them either in the absence or presence of ethanol, and then we treated them with various amounts of these recombinant cytolysine subunits. And you can see here that these are the untreated primary hepatocytes, and then we added like 1 subunit of the large cytolysine subunit, and you cannot see any cytotoxicity, or at least a baseline cytotoxicity. And then when we add the 2 subunits of the cytolysine, all of a sudden we can increase cytotoxicity. And in particular, if we add the highest dose of cytolysine subunits in an equimolar dose, you can see that this is a significant increase of cytotoxicity as compared to the untreated hepatocytes, suggesting that the cytolysine is directly hepatotoxic to the liver. So now we wanted to move back and say, you know, what would happen is, or we wanted to compare, is this also true in our patients with alcoholic hepatitis? And now what we did is we used germ-free mice. So germ-free mice are completely devoid of any bacteria, of any fungi, and of any viruses. So they have no microbiome present. And then we colonized these germ-free mice with either stool from cytolysine positive patients with alcoholic hepatitis, or with stool from cytolysine negative alcoholic hepatitis patients. And then we fed these mice again chronically alcohol. And the results are shown in the next slide. When we did this, the groups that were colonized with feces from alcoholic hepatitis patients who were cytolysine positive had the highest liver injury, highest liver steatosis, as compared to the groups that were colonized with stool from cytolysine negative patients with alcoholic hepatitis. And most importantly for us, what we were able to detect now is that in the mice that were colonized with the stool from the cytolysine positive alcoholic hepatitis patients, we can now detect the cytolysine, in fact, in the liver from these mice. We obviously cannot detect them if the mouse was colonized with stool from cytolysine negative donors. And also, quite intriguingly, if you don't feed them alcohol, even if they have cytolysine in the microbiome, this does not make it to the liver. So suggesting that ethanol somehow is required, that this cytolysine, and especially what we currently believe is that this bacteria can move to the liver and exert some damage there. So what I have shown you so far, and hopefully convincingly shown you, that cytolysine seems to be a very good biomarker for alcoholic hepatitis patients. It seems to be sufficient to cause some mild liver disease, and especially to exacerbate alcohol-induced steatohepatitis. Now, to demonstrate that cytolysine is in fact necessary for the disease, we have to specifically either eliminate cytolysine, or in our case, what we try to do is we have to eliminate the cytolysine producing Enterococcus faecalis. And to do this, we turned our attention to bacteriophages. So phages are are viruses, very small viruses that are very specific to one particular bacterium. You can see various forms of phages. They all have a head and a tail. With this tail, they can inject their DNA into the bacteria. Inside the bacteria, these phages can replicate. They can make enzymes to destroy the bacteria. The bacteria will be dead subsequently, but the phages have been replicated inside the bacteria, and they are now released to the outside where they can infect more bacteria and kill more bacteria. So we isolated now phages that can specifically target the cytolytic Enterococcus faecalis. You can see one of the plaque assays as an example over here. And then we obviously also characterize these phages. In this case, I'm showing you electron microscopy image from our cytolytic Enterococcus faecalis bacteriophages. And that the experiment that we have chosen to use is, again, we used germ-free mice. We colonized, in this case, all of them with feces from cytolysin-positive alcoholic hepatitis patients, and then we treated them either with a control phage. In this case, the control phage was directed against the bacterium C. crescentus that is not present in the mouse or in the humans, and we used obviously our cytolytic Enterococcus faecalis phage. And when we did this, you can see the results over here. So these are one group of mice that were colonized with the same donor from a cytolysin positive alcoholic hepatitis patients. This is a mouse group that was treated with the control phage, and this is a mouse group that was treated with the phages directed against the cytolysin positive Enterococcus faecalis. And you can see that we can achieve a significant decrease of liver injury, significant decrease in hepatic steatosis. And we have replicated this in a different mouse group colonized with a different donor who was cytolysin positive. And most importantly for us, what we have also been able to achieve by this bacteriophage treatment is that we can also decrease the cytolysin, so the toxin in the liver that can directly kill liver cells. So in summary, what I have shown you now is that ethanol somehow, and we do not understand the exact mechanism, in some percentage of, or in a certain subpopulation of alcoholic hepatitis patients with very severe disease, they have a higher colonization. These Enterococcus faecalis can migrate and translocate to the portal vein, where they can either dock onto TLR2, and we have shown this in our previous publication, which induces liver inflammation, in particular IL-1 beta, which seems to be very important for liver disease. And this drives directly hepatocyte steatosis. But then what we have shown now is that cytolysin is also present in these mice in the liver, and cytolysin can directly kill hepatocytes and cause a disease progression, at least in our mouse model. And the novelty, obviously, that we have, that I've shown you now, is that we have bacteriophages that can very specifically now target this cytolytic Enterococcus faecalis. They can kill this the bacteria. They can decrease cytolysin production and can ameliorate liver disease. And this was, very much highlighted in the scientific literature. And essentially, this was praised for, now we are able to edit very precisely the gut microbiome. I want to end this talk by just showing my very enthusiastic, very dynamic and young group of people in my laboratory, and in particular, Cristina Laurenti, who is the mastermind behind cytolysin, Eduan, who did most of the work with the mouse models that I've shown you today, and also Sonia Long, who did most of the clinical analysis. Lu Jiang, she did most of the fungal microbiota work, and obviously all my funding agency. I just want to show you and invite everybody who is interested in alcohol-induced end organs damage. This is a basic translational but also clinical conference that myself and Dr. Sabo are organizing, hopefully in person next year in Ventura, California. So if you are interested, please come and join us. we have secured a keynote speaker, Bruce Beutler, who received the Nobel laureate, who is a Nobel laureate 2011 for discovering TLR4, which we know is very interesting, very important for alcohol-induced liver disease. I would like to thank you for tuning in and for joining me today for this talk. And I believe we have now question and answer session. Thank you.
Thank you, Professor Schnabl. I think this was a very enlightening talk and something new happening. And I think these are going to be the newer aspects that we will be seeing in the next decade, or maybe earlier we will have some advanced therapeutics in this area. I think it has been interesting. We heard from Dr. V.G. Mohan Prasad on the on microbiota in health and disease. I think Dr. Vineet covered inflammatory bowel disease and then now liver disease. So this has been very exciting. Before we go on to the question answer session, we have Dr. Abhi Phillips with us, and he's a young researcher from India, has done a lot of work and has shared that he has done and how it looks as of now. So, Dr. Abhi Phillips, are you on?
Yeah, thank you, ma'am. I'll be speaking a couple of minutes on some recent work that we had done on the microbiome. And then I think we can go on and move on to the question answer sessions from the audience. I'll just share my screen. So is this visible to all?
Yes, Abby, it's visible. Yes, you can go ahead.
Yeah, so I mean, exactly what Professor Schnabl, he is the authority in actually defining the basic science behind what we see in alcoholic liver disease. So what we have actually done is that we might have jumped the gun a few years back by doing a pilot study, but on FMT and how that actually modulates the microbiota, leading to better outcomes in these patients. But what actually we need to understand is that why is this happening and how can we actually improve our understanding of gut microbiota, because it is not same among each patient in the same region, and it's all different in other regions also, out of the country, in other continents also. So what our work currently is to ideally look at what is happening in patients with alcoholic hepatitis who have severe disease versus a mild disease. And this is one of the papers that we have currently in peer review, where we actually show that patients who have died because of severe alcoholic hepatitis vis-a-vis those who survive, we have shown that specifically this is a Circos plot, and this is the multivariate analysis using DEFC method. We have shown that there are specific species that are attached to these events. Like you can actually look at the genera that can that are actually abundant in patients with alcoholic hepatitis who die versus the ones who actually survive. Now, when we looked at these genera specifically and their interactions using a network analysis, we found out that these genera actually interact with each other in a positive or a negative way. For example, we can see that this particular Enterococcus, which is very commonly seen in our alcoholic hepatitis patients also, the Enterococcus species was actually beneficially or positively interacting with another particular genus. And possibly what Professor Bernard Schnabl was telling us, why is this happening, is possibly because the cytolysine theory of it is possibly because there is definitely interactions between these genera or the genus, each genus in the patient, that actually leads to that outcome where some patients have high cytolysine or some patients have some other toxic product that is actually leading to a more disease severity. So this is just basic work we have done in 25 to 30 patients, and this actually shows that it's not only a particular species, it's actually the species interaction with each other and the metabolite that they produce, which actually can cause the disease severity. Similarly, when we looked at patients with and without sepsis, we actually found out that in patients with different sites of sepsis, for example, you look at patients with bacteremia versus patients with peritonitis in cirrhosis with alcoholic liver disease, we found out that there were different groups of bacteria that were significantly altered in patients where there is infection based on different sites of sepsis. Even the inflammatory marker. So we have patients with very high interleukin levels. So this is interleukin 6 that we have identified. And in patients where interleukin 6 was very high, we found out that organisms that were actually associated with severe immune exhaustion were actually positively high, abundant, rather than the other ones which are actually much, much lower in patients without a severe inflammatory response. Now, all of these are important because most of this is driven by the metabolite. It is not just the family or the firmicute part or the species part. It is actually interactions between these group that actually produce these metabolites, and these metabolites are the ones that actually lead to disease worsening or disease causation, which we still have to identify. So this is the summary slide that we had submitted for peer review it's under peer review at the moment, where we actually show that hepatic encephalopathy, acute kidney injury, even treatment-related outcomes, that is steroid versus growth factor versus abstinence, and presence of or absence of infections, even the liver disease severity based on the MELD and CTP cutoff, everything has a specific group of bacteria attached to it, which promotes it or either reduces the severity. So in that sense, the important part is that it's not just the bacterial genera, but actually the dominance, the authority, and influence each other has on themselves or the counterparts within the gut that actually decides the disease status and the disease severity in a patient with alcoholic liver disease. Now, this might be even true for patients with other diseases also, but I think it's a matter of further research and future research. And I think a lot of such good data can be generated from basic science lab, like what Professor Schnabl is working on. Over to you, madam, for taking over. I'll stop sharing my slides now.
Thank you. Thank you, Abby. I think that was really an insight into the work that is being done. And I'm sure in future you will be able to give us more insight and more in-depth analysis on the same. There are a lot of questions coming actually, and I will direct a few questions for want of time. The first question for Professor Schnabl is that from Dr. Vamsi: in acute alcoholic hepatitis, is it possible to culture the cytolysine positive Enterococcus in the blood sample? And there is a second question related to the same, that is, can antibiotics against Enterococcus provide benefit like bacteriophages? Any effect or evidence of effect of antibiotics on Enterococcus? So 2-3 questions on the same issue.
Thank you so much. I think these are excellent points. So can we decrease the cytolysine positive Enterococcus faecalis in the intestine by simply antibiotics? I think I heard that some trials are being started about using antibiotics against gram-positive bacteria. We will, as far as I know, we will hear already one at AASLD in November at our liver meeting. I do not know the outcomes, but as far as I know, we will hear the results. Unfortunately, this study has not collected human stool, so I do not know if cytolysine was altered depending on the outcome, what we will see. But I think it's fairly there's certainly a possibility that antibiotics might be helpful. What we have just published in a very nicely is that what also seems to be, at least in the mouse model, we have not looked so much into patients with alcoholic hepatitis, that the glycocalyx, so this sugar composition in the glycans inside the intestine, seem to be changed in patients with alcohol use. And that probably predisposes like more of the pathobionts to flourish. and to suppress some more beneficial commensal bacteria. So there might be actually avenues where we just use like very targeted prebiotics. And prebiotics, we have a lot of noise about prebiotics, but I think if you have very targeted, that we can use probably prebiotics that we know that they, suppress the adhesion or the colonization of this cytolysin positive Enterococcus faecalis. I mean, that could be also certainly a good avenue moving forward.
Yeah.
Thank you, Dr. Schnabl. Dr.
Rupa,
Vineet.
Rupa, can
Yes,
I ask
please, yes.
Bernard a question on this? Both Abby and Bernard a question on this. Like Abby, you also showed the interaction between the various genera and the species. So when we look at the case of Akkermansia mucinophilia, so that's a very specific case in terms of, at times, when it is increased, it may show some benefit. Whereas in another disease, it may not show any benefit. So if you use that example and juxtapose it on the networking of the various phyla with each other, what do you think? Like, how do you think you'll be able to explain that?
I think Professor Schnabl, you can give us some insights on that.
No, I completely agree. So with this Akkermansia, I'm also a little bit puzzled, to be very honest, because it's almost like a hype now about Akkermansia. But I mean, Professor Tilk from Austria, he showed very nicely that that in alcoholic hepatitis patients, we actually have confirmed this in an independent study that at least Akkermansia is depleted also like in patients with alcoholic hepatitis, and at least when he gives it back to in a mouse model, there seems to be some benefit. But I completely agree. I mean, in a different disease context, it might be completely different. And obviously, these are mouse studies. We have to be always very cautious. Can we really translate this directly into humans? So that's always the question. But I completely agree that this might be, disease context specific.
Yeah. So I mean, why does Akkermansia behave like this? I think it has to do with what Akkermansia is actually doing along with others around it. And that's very important. The bacterial interactions, I think, are more important than specifically a single bacteria, because we have Klebsiella and E. coli abundantly in our bodies, but we don't actually see them cause a disease unless and until something is sparked. For example, in alcoholic hepatitis, Enterococcus faecalis is one of the most important genera that can actually cause severity in the liver disease. Similarly, I think there is a spark. There is something that is happening in case of alcoholic liver disease or inflammatory bowel disease, where potentially beneficial organisms are being subdued and pathobionts are forming because of specific interactions. And these interactions may be direct interactions, or it could be interactions through metabolites. And I think it's all part of what we have to find and look into the future. And we don't have any answers to these at the moment.
I think across organs, it's the same thing. There is dysbiosis causing disease, and we are just still looking into what will be the best option to correct this dysbiosis. And that is where we go. That's the future, I suppose, from what we are hearing. Dr. Vineet, there is a question in fact, a lot of questions for you as well, but the Indian questions from Dr. Vimal B. Saradeva. He wants to know about the diet. Does regular curd in diet, how does it help? How does turmeric help? If you have any insights on whether this works via the microbiome, or how beneficial and why they are beneficial.
So curd should help, because in the sense that curd is what Mechenkov, the person who won the Nobel Prize in 1907 and the one who came up with the probiotic concept, showed that Bulgarians, they had the maximum longevity to life because of curd. So curd should definitely help. There's no doubt about that one. And again, if we look at the probiotics, they are all based in a way on the fermentation process and the curd and all that. Second is curcumin. Curcumin is pleiotropic. It has like anti-inflammatory effect, anti-cancer effect. It has a lot of effects. So if I look at the trials which have been done on curcumin starting from 2000 to 2020, then somewhere around 2005, we looked at oral curcumin, found it not so useful because we did use less doses. Then 2009, we had curcumin enemas, which we found to be very useful. 2020, we have a trial recently published by Rupa on nano formulations of curcumin, which Rupa can talk about, and its effect on ulcerative colitis. So curcumin is like the grandmother's remedy. Like in India, the grandmother's Dr. Schnabl, there's a grandmother's remedy. Anybody gets hurt, they put a paste of curcumin and put it on that wound. Or they'll put curcumin in milk and give it to it. And that in a way has benefited probably. We may not be able to gauge the magnitude, but so curcumin has a signal, but how specific that signal is, we are not very sure, but it certainly doesn't do any harm. What do you say, Rupa? You just published a trial on it, so you can also talk about it. I think Rupa's signal is...
I think Rupa's signal is down. So
curcumin again, like basically Rupa's trial was nano formulation of curcumin, and she found that it was useful in patients with ulcerative colitis, because the problem with curcumin is the bioavailability. If you give it in the oral raw form, yeah, Rupa, you can talk about your trial.
Yeah. I'm sorry, we are having some connection issues. I think turmeric, of course, there's no studies, Vineet. Is there on microbiome alteration with turmeric? What we actually did was we identified a more bioavailable form of turmeric. It was useful as an additive therapy to remission and maintenance of remission for ulcerative colitis. The Israel group also have found similar results. So turmeric perhaps acts, but I'm not sure exactly how via the microbiome. Any insights onto that?
So I think what curd is to microbiome, that curd probably gives a benefit, but nobody has gauged the benefit. Similarly, turmeric is to inflammation. It probably takes care of inflammation, but nobody has gauged.
And to Vineet, I just wanted to add here that if you combine piperine with turmeric, it enhances the absorption of curcumin 1,000-fold. So I think some of the novel formulations are a combination of piperine in small doses, like 1/100th of the dose of curcumin. Then it works, probably improves the absorption. So any trials that you're using in a combo of piperine and curcumin to Vineet and Rupa?
So the one that we are using, Dr. Vijaya, is actually not a combination. It is extract of the curcumin proper and getting out the bioavailable molecule from it, using a technology whereby it becomes more lipid soluble, thereby enhancing things. So we have done, yes. So lesser doses may be effective. But regarding the mechanism, as I said, via microbiome, maybe via microbiome, but I don't see many studies so far in that area as to how exactly it works. Anti-inflammatory is what we all know, and it's a panacea now. They're used for everything. But in ulcerative colitis, there seems to be an indication that perhaps it will be a safe add-on to regular therapy. So in the want of time, there are a lot of questions coming. Dr. VGM, I think you should answer this. There is a question coming.
No, there are a couple of quick answers from Vineet on super donor. Also, Professor Schnabl can also talk about it. Who's a super donor? So can you define a super donor at this point of time?
So basically in the FMT trials, it was seen that there were certain donors which could sway the result of the trial, in the sense that the patients would benefit a lot when transplanted from that specific donor. Analysis of those donors showed that, okay, they had maybe higher diversity, richness, and homogeneity and all those things. So the term came up super donors, but what are the criteria for super donors has not been established. That is an area of what I would say active research at the moment in the field of microbiome. And if anybody can come up with 1, 2, 3, 4, 10 commandments for the super donor, that I would say would be the biggest breakthrough for this year or the next year.
Professor Schnabl, could you also
add?
add?
You have to find an intelligent person. the microbiota of a Harvard guy is different from that of the general population. So, identifying the ideal donor and the super donor is going to be very, very crucial. There is another quick question coming for Professor Schnabl. This is from Ela Srinivas from Hyderabad. Any role of antibiotics or probiotics in treating enterococcus in animal studies in preventing NASH? So any role of antibiotics
very good.
via the same mechanism in preventing NASH?
Yeah. So NASH, we have, I'm a translational person, so I usually start in patients and then move to animal models. In NASH, we had a very, we just published this earlier this year. We had like 100 patients with NAFLD and various stages of NASH, so even NASH cirrhosis. And there was no signal for Enterococcus faecalis at all. And also there was no cytolysin positivity. So in other words, this seems to be, at least as far as we know right now, this seems to be very specific for alcoholic hepatitis, but not for NASH.
We need a quick question for you from Dr. Sanjeev Khanna, Mumbai. Does a plant-based high fiber diet, everybody wants to know about diet and microbiome, does a plant-based high fiber diet lead to better bacterial diversity compared to a meat-based non-vegetarian diet?
Yeah, it does. It does. In fact, there was a study which came up in Nature 3 years back, I think. Simple study. Volunteers taken 5-day vegetarian diet, subsequently 5 days non-vegetarian diet, and it clearly showed that there was a difference in the microbiome. Within 5 days, the microbiome changed. Now, if you go to Japan, there's a group which is led by Dr. Chiba. They have come out with 10 or 15 consecutive studies on plant-based diet in patients with ulcerative colitis and Crohn's disease. And as the group believes that plant-based diet has actually led on to revolutionary results in inducing remission in patients with ulcerative colitis. This is that kind of group. The way you have the group in Australia which works on FMT, so similarly this is the group in Japan which works on plant-based diets, Chiba's group, and showed beautiful results. So it has merit. There is no doubt about it. And that's why we talk about the rural microbiome, as I showed you the study, is better than urban microbiome. That is why you say that although IBD is coming up in Africa, but if you go back 5 years back, if you want not to have IBD, you should migrate to Africa.
Wow. There is a quick question from Dr. Vimal Saradeva from Rajkot. He's asking on the CRAP capsule. Is there a FMT capsule available in India? Will it come in India? It's available in the US, I'm sure. How expensive?
Open biome, it comes in.
Open biome is actually using frozen fecal capsules and providing it for research purposes. They're actually known as capsules. I mean, in simple terms. But I'm not sure if any company is actually looking into a proper stool-based capsules. But like Professor Bernd Schnabl was saying, they're looking at a newer generation of bacterial metabolite or a newer generation of mixtures of bacterial live bacteria in capsules, but not for not like a
manufacturing by the pharma will take a big totally new different area, because we were talking into development of these capsules. Now what happens for the pharma also is that they need a separate biolab. The regular pharma area cannot be used for making these kind of capsules. So for any pharma to go into it will take time. Yeah, yes, Vineet.
Yeah, Abby, the thing is that Dr. Sareen is already into a project with IMTech. From last year, the project is on, whereby they are looking at making microbiome-based capsules and all, right? And at the same time, I showed you that is for India. At the same time, I showed you a slide where there was a table of various food trials which are going on already, and those are like biotherapeutics as well as bacteria-based capsule as well as metabolite-based capsule. But India also the process has started already.
I think Rebiotics is one company that is actively looking into spiferring, trying to make capsules out of live bacteria. rebiotics. They have not come to Asian continents yet, but they're still in Europe and the US, I think.
If the moderator would permit, I have one last question on Mycobacterium avium intracellular paratuberculosis, the MAP. And what do you think? How many Crohn's patients would have MAP in India? What do you think? Or even in the US? What percentage of Crohn's patients it is because of MAP?
We did many years ago. We did a small study. We actually did not find any in Crohn's patients, but I suppose that was a long time back. I think almost 7-8 years back. We published that. We did not find any association.
So that's a very interesting question. Few years back, we published whereby we looked at MAP, because in the sense that India has high incidence of Crohn's disease, India
Yes.
has high incidence of tuberculosis,
Closest.
India has high incidence of Johne's disease. Johne's disease is a disease caused in ruminants by MAP. So a few years back, we felt that probably in a small study that MAP was in Crohn's disease was there. It is not that it was not there at all. It was probably there. Then we embarked on a multi-center all India study where over 15 centers, we just completed that study, where over 1,000 patients have been recruited, and of which like these are patients of Crohn's disease, ulcerative colitis, tuberculosis, to look at it. So what I can tell you is the preliminary results of that data. The preliminary results of that data clearly show that in India, although MAP is high, yet at the same time, Crohn's disease, the percent prevalence of MAP was not so high, and it was done by 2 or 3 multiple methods like in situ hybridization and MAP RNA and all those things, MAP DNA and in situ hybridization. So maybe in a year's time or something, the results should be in print or something.
So I think we've had an exciting discussion. We'll end with a quick one-liner from Professor Schnabl and Dr. Vineet. When do you think this is going to come to clinical practice? What do you think, Professor Schnabl?
I think we have,
I mean, is it 5 years? Is it 10 years?
I think we have a long way to go. And I think one of the most important things is I do not believe. I think we just wrote a review in Cell Host and Microbe and made this very clear. I think not each patient is driven by the microbiome. And I'm not an IBD expert, but I guarantee you, you have a subgroup of patients who's driven more by the microbiome rather than by autoimmunity or something like this. Definitely in the liver disease field, we cannot, say that every microbiome therapy will be useful for every NASH patients. Not every microbiome therapy will be useful for any of the PSC patients or so. I think we have to personalize this approach. We have to use personalized diagnostics, as I have shown you. And, and then we can start thinking about microbiome-based therapy. I think this is my bottom line, that we have to really use, first of all, personalization, and then precision medicine for therapy. So that's my hope for the future.
Abby, you want to have
Dr.
a take
Vineet,
at it? Okay, what I would say is that I fully agree with Dr. Schnabl. Microbiome is one of the players. It's like a monarchy, or the ministry, where you're the prime minister and you have 4 ministers. So one is the genetics, the one is the microbiome, other the immunology part. Until now, it's very clear that microbiome is not the only culprit. Yet at the same time, it does have its role plays its role in share of crime. So you need to give it that much importance. And as he very rightly said, it will have to get a little bit more personalized. But that's where the field is going to go.
Dr. Mohan Prasad, your comments, please.
No, I think I would
Dr. Abby as well, yeah.
to comment, and then finally we will come in to thank all of you. Abby.
From the lively talks that we had, I think, this is just like social media, the gut microbiome. You have influencers, you have authoritarians, and then you have these broadcasters. They don't influence, but they broadcast. That is what is happening in the microbiota. I mean, especially with the bacterial population, I believe, and we are yet to understand how they do this broadcasting, how they influence, how they authoritize others. So this is very important in identifying personalized therapy in the future. So, like, this is the most important part what Professor Schnabl has done with his Enterococcus faecalis and cytolysine study, and that is exactly what we should be doing, and that is the kind of work that we should be developing from India also, to see in our population what is going to be the personalized options for our patients.
I think it will be our dream to work out the genetic makeup of all the microbiome in every individual's gut. Probably find what is the bad, microbiota or microbiota, or even the viruses, and then try to specifically treat them with the live biotherapeutics. Probably it might take at least a decade more for us to achieve this. That's what I guess. So,
Depending on the funding that we receive, funding is a big part of the
Yes,
project.
I think that's yes.
That's the unseen part. Actually, it's very important.
thank you so much, Rupaji. So, should we call it a day? I just will request Dr. Vamshi to thank all the speakers. Our special thanks to Professor Schnabl, and apologies that you had difficulty in logging in at the beginning. So, luckily, everything got sorted out. And thank you so much and to Professor Vineet, who had to run in from another webinar. So, and to Abby, who was instrumental in getting Professor Schnabl here, and to the great lady Rupa. So over to Vamshi for a formal vote of thanks.
Well, it's a privilege to propose vote of thanks on behalf of BGM Foundation and in association with AIG Hospitals, Hyderabad. Thanks to all the stalwarts for making this very interesting Sunday. Thank you, Professor Schnabl, Professor Vineet Ahuja, Professor Rupa Bane, Professor VGM, and upcoming star, Dr. Abby. Though each and everyone are very, very far away physically, we could all connect today digitally. And this was possible because of our live streaming partner, Imagica Health, and our educational partner, About India. Thank you all for all the support, and thank you all delegates for taking time on this IPL Sunday evening and viewing this webinar. So we will sign off today and hope to meet you soon.
This lecture all the lectures will be posted on our VGM Foundation website. So the viewers probably can go back and listen to these lectures. Thanks to Arbot for supporting us on this platform. Thank you.
Thank you very much. Thank you so much.
So we're just signing out. Thank you, Professor Schnabl.
Thank you.
Thanks, Abby and to Rupa and to Vineet, sir. Thank you. Bye-bye.
So thank you all.